So this is my presentation about the representation of a primary article within a news article. My name is Kara Barth, and I'm in Bio 282 Conceptual Approaches for Biology Majors 2 at Arizona State University for spring 2021. So the article background. The title of the article is HIV-like virus edited out of a primate genome, coming closer to a cure for human HIV infection. It was released on November 30th, 2020. And it was sourced from Temple University Health System, specifically Lewis Katz School of Medicine, which goes towards saying that it might be a reliable article. CRISPR technology is mentioned a lot throughout the article, so I added the photo on the left just to show what it is. It shows what CRISPR actually stands for. Essentially, CRISPR goes in and can edit the DNA within a genome. So it can add or delete parts of the DNA that could be mutated or missing. So the article summary. The article goes in a lot about the interviews between the interviewer and Dr. Kamel Khalili and Dr. McLean. So Dr. Khalili appears to be the lead of the investigation according to the article. So the article then goes in to explain the similarities between SIV and HIV, which is essentially the same thing, except that SIV occurs within primates and HIV occurs within humans. It then explains how the technology worked to cleave the DNA and goes into how the experiment was laid out. It specifically mentions that 3 random infected macaques were chosen and injected with the CRISPR technology, and that there were 3 controls, and that after 3 weeks, they harvested tissue and blood to see the results. The article goes to claim that the treatment was highly effective and that both scientists are very hopeful that this treatment could be good for HIV. It then talks about Dr. Khalili and her 2020 Nobel Prize in Chemistry. So the primary source. It was authored by many people. As you can see, I listed them all there. However, you can see in there that Kamel Khalili and McLean are listed, so they were actually authored. The actual primary source was published on November 27, 2020, and it again goes into CRISPR technology, but it also goes into ART technology. So I added on the left what ART stands for. It's antiretroviral therapy, and it reduces the amount of HIV in the body. So essentially, ART technology goes in and it reduces the HIV viral load within the body, which suppresses the symptoms of HIV. However, it's not a cure, which is why the CRISPR technology is being tested and looked at. It just suppresses the symptoms for a little while. So the technique. Adeno-associated virus is released into the primate, and it targets all of the tissues and spreads out far and wide. And it expresses the Cas9 endonuclease and guide RNAs to a high extent. It also uses gRNase Cas9, which is essentially CRISPR Cas9 but modified specifically for this experiment, and it allows for the editing and removal of viral DNA between the cleavage sites from the host genome. Essentially, CRISPR Cas9 cleaves and removes SIV fragments. It does this specifically in places where the SIV virus is in higher loads, which happen to be the lymph nodes, spleen, bone marrow, and brain, just to name a few. So the primates were injected with the virus. They were then given the ART treatment for 2.7 months, and then 3 of them were randomly chosen to be given the CRISPR treatment, while the others were left as controls. So these were the results. The first graph shows the SA Cas9 expression within the primates. Down below, you can see all of the places where the expression was recorded. So at these places, like I mentioned before, are places where the SAV virus would be higher. So this is essentially going to show that the Cas9 did target those places where the SIV virus was higher. And again, results. So the first line is the red line, which is the untreated primate, and the other 3 lines were the 3 treated primates. So this is prior to the treatment and after the treatment, the percent of proviral DNA in the lymph nodes, which again, as I mentioned before, the lymph nodes had higher SIV loads. So this is showing how the treatment worked and if it worked well in these places where the viral loads were higher. So, the accuracy of the article, yes, I do think that the article was accurate. It correctly describes the methods for going through the experiment as well as the techniques used in the experiment, and it also credits the main scientists. It is very brief and oversimplified, though, so I don't think it was the best, but it did do its job well. It couldn't be that much longer. And it doesn't mention the art treatment, which I do think would have been good considering it was within the experiment and it is the basis for HIV and SIV treatments currently. And these are my citations.